Oncology Clinical Trials Endpoints. Introduction. Endpoints and outcomes in oncology clinical trials are critical components that determine the success and efficacy of the investigational treatments. These metrics provide a basis for evaluating the therapeutic benefit of a new treatment, its safety profile, and its overall impact on patients' health and quality of life. Here's a more detailed look at the different types of endpoints and outcomes commonly used in oncology clinical trials. Endpoints. Primary endpoints are the main results that the clinical trial is designed to measure. They provide the most important data for evaluating the primary objective of the study. Secondary endpoints provide additional information about the treatment's effect. Exploratory endpoints are used to gather additional data that may not be central to the study's primary and secondary objectives but can provide valuable insights. Surrogate endpoints are indirect measures used to predict clinical benefit. They can expedite the evaluation of new treatments but must be validated to ensure they accurately reflect meaningful clinical outcomes. Primary endpoints. Overall survival (OS) measures the time from randomization or start of treatment until death from any cause. It's considered the gold standard for assessing the efficacy of cancer treatments. Progression-free survival (PFS) measures the time from randomization or start of treatment until disease progression or death. PFS is often used in trials for cancers that are difficult to treat or in cases where OS may take longer to measure. Disease-free survival (DFS) similar to PFS but specifically measures the time after a curative treatment, for example surgery, until the recurrence of cancer or death. Objective response rate (ORR), the proportion of patients with a measurable reduction in tumor size, partial or complete response, based on predefined criteria such as RECIST, response evaluation criteria in solid tumors. Secondary endpoints. Quality of life (QOL), assessed used patient-reported outcomes (PROs) and questionnaires to measure the impact of the disease and treatment on patients' daily lives, physical functioning, and overall well-being. Duration of response (DOR) measures the length of time that a tumor continues to respond to treatment without growing. Time to progression (TTP), the time from the start of treatment until the cancer progresses, not including death from other causes. Safety and tolerability assesses the frequency and severity of adverse events, side effects, and treatment-related toxicities to evaluate the safety profile of the treatment. Exploratory endpoints. Biomarker analysis. Identifying biological markers that predict response to treatment, resistance, or adverse effects. Biomarkers can include genetical mutations, protein expressions, or other molecular characteristics. Pharmacokinetics (PK) and pharmacodynamics (PD), studying how the drug is absorbed, distributed, metabolized, and excreted (PK), as well as its biological effects on the body, which is PD. Immunological response, particularly relevant for immunotherapies. This endpoint assesses changes in immune parameters such as levels of immune cells or cytokines. Surrogate endpoints. Tumor shrinkage. Early tumor size reduction can be an indicator of treatment efficacy but must correlate with long-term outcomes like OS or PFS. Circulating tumor cells (CTCs). Changes in the number of CTCs can indicate treatment effectiveness and disease progression. Composite endpoints. Composite endpoints combine multiple individual endpoints into a single measure, providing a broader assessment of treatment impact. Event-free survival (EFS) combines several adverse events such as disease progression, relapse, or death into one measure. Clinical benefit rate (CBR) includes complete response, partial response, and stable disease, offering a comprehensive view of treatment efficacy. Importance of endpoint selection. Regulatory approval. Endpoints must meet the requirements of regulatory agencies like the FDA or EMA for new drug approvals. Clinical relevance. Endpoints should reflect meaningful benefits for patients such as improved survival, reduced symptoms, or enhanced quality of life. Statistical power. Choosing appropriate endpoints ensures the trial is adequately powered to detect significant differences between treatment groups. Comparability. Standardized endpoints allow for comparison across different studies and meta-analysis, aiding in the broader understanding of treatment effects. Conclusion. By carefully selecting and rigorously evaluating these endpoints, oncology clinical trials can provide robust evidence on the efficacy and safety of new cancer treatments, ultimately guiding clinical practice and improving patient outcomes. Please like, share and subscribe. Leave a feedback comment mentioning any topic that you want me to make a video. Looking forward for a positive response. Thanks for watching. Please like, share and subscribe. Leave a feedback comment. Many thanks for your support and having great connection.